History of Life on Earth. Chapter 3, The Eukaryotic Cell. The merger that made everything else possible. 3.1 It happened once. Chapter 2 ended with a swallow. One cell surrounded another, somewhere in an unremarkable stretch of ancient sea, and failed to digest it. In the long history of the ocean, this must have happened an uncountable number of times. Cells eat cells. It is the oldest business in the world, and a failed digestion is nothing to write home about. But this time, the meal stayed. And as far as anyone has been able to determine, and this is one of the most extraordinary claims in all of biology, this particular merger, the one that produced the complex cell, left exactly one surviving dynasty. In the ancient oceans, similar mergers may have happened thousands of times. Cells swallowed cells constantly. But only once did the partnership stabilize, balance its books, and endure. Not once per continent, but a single lineage that outlasted all others in 4.5 billion years of planetary history. Every organism you have ever seen with your unaided eyes is descended from that single accident. Every tree, every mushroom, every fish, every insect, every bird, every person. Trace the ancestry of a blue whale and the ancestry of a redwood back far enough, and they meet at one cell that swallowed another cell and didn't finish the job. And if it hadn't happened, Earth would still be alive. It would still be beautiful, stromatolite domes in the shallows, mats of purple and green slicking the rocks, an atmosphere breathing in and out on a 10,000-year cycle. Chemistry humming along exactly as it had for 2 billion years already. It just wouldn't have anyone in it. No forests, no eyes, no wings. No one at all to stand on the shore and wonder how any of it got here. The reason there is anybody to ask the question is a digestive failure. 3.2 The Tent and the Cathedral. To feel the size of what happened, you first have to see what a prokaryote actually is. It is small, 1 to 5 micrometers, which is to say you could line up a couple of hundred of them across the width of a human hair. While many bacteria organize their interiors with clever protein compartments and internal tracts, it has nothing like the sprawling architecture of a complex cell. Its DNA lies loose in the cytoplasm unfenced. There is largely one main room and everything happens in it. Reading genes, building proteins, generating power, sensing the world, dividing in two. It is a tent. This is not an insult. A tent is a superb piece of engineering. It is light, cheap, fast, and can be pitched almost anywhere, which is precisely why bacteria and archaea have colonized boiling springs and Antarctic ice and the rock beneath your feet. And why they will still be here long after we are not. Now look at a eukaryotic cell. It is 10 to 100 micrometers across, and don't let that fool you, because volume grows as the cube of length. A typical eukaryotic cell holds thousands of times more space inside it than a bacterium. If the bacterium is a tent, this is a cathedral, and the cathedral has rooms. There is a walled library at the center, the nucleus, holding the genome behind a double membrane with guarded doors. Radiating out from it is a vast internal folded landscape of membranes, workshops, sorting rooms, recycling yards, a courier network. Scattered through the interior are hundreds or thousands of mitochondria, the power plants, and, as we're about to see, they are not really part of the cell at all. Running through everything is a scaffold of protein cables, constantly assembling and dissolving, along which molecular porters physically walk, hauling cargo. And the genome is enormous by comparison, not just bigger, but structured, packaged, regulated, layered. So here is the question this chapter has to answer. You do not get from a tent to a cathedral by slowly letting the tent out. Something else happened. 3.3 The woman who was right. In 1967, a young biologist published a paper arguing that the eukaryotic cell is not one organism at all. Her name at the time was Lynn. She was married to the astronomer Carl Sagan, and she would later publish under the name Lynn Margulis. Her paper was reportedly rejected by something like 15 journals before the Journal of Theoretical Biology agreed to run it. Her claim was this: the mitochondrion is not an organ of the cell. It is a prisoner. It was once a free-living bacterium, swimming and feeding and dividing on its own account, until it was taken inside another cell and never released. The same, she said, goes for the chloroplast in plants. The complex cell is not a creature. It is a federation, a merger of separate organisms that stopped being separate. The idea was not entirely new. Versions of it had floated around at the turn of the 20th century and been quietly dropped. What was new was Margulis's willingness to defend it in detail, at length, and against a wall of professional derision. The reception was, by all accounts, brutal. The idea was regarded as fringe, and so, for a while, was she. Then the sequencing technology arrived, and the molecules were read, and she was right. Not partly right. Not right in spirit. Right. A footnote in fairness, because this is how science actually works. Margulis went on to make other endosymbiotic claims, that cilia came from captured spirochete bacteria, among others, that the evidence has not supported, and she held other positions that the field rejected. Being spectacularly right once does not confer permanent correctness. It just means you should be listened to carefully, which is all anyone is owed. 3.4 The evidence is inside you. The case for endosymbiosis is not an argument. It's a pile of fingerprints, and they're in your body right now. They have their own DNA. Your nuclear chromosomes are long, linear, and wound around spool. Proteins. Your mitochondria carry small, circular loops of DNA untethered by vast nuclear scaffolding, which is exactly what a bacterial chromosome looks like and nothing like what a eukaryotic chromosome looks like. They have their own ribosomes. And those ribosomes are not built like the ones out in your cytoplasm. They're built like a bacterium's, which is why some antibiotics make you sick. Several important antibiotics work by jamming bacterial ribosomes. They also, to a degree, jam mitochondrial ribosomes, because those ribosomes are close enough to bacterial that the drug can't tell the difference. Some of the toxic side effects of these medicines come from precisely this. Your mitochondria are still bacterial enough to be poisoned by drugs designed to kill bacteria. They have two membranes, an inner one and an outer one, with a gap between. The classic interpretation writes itself. The inner membrane is the bacterium's own. The outer one is the bubble that wrapped around it. While biologists still debate whether that outer layer came from the host or the bacterium's own outer skin, the result was a double-walled fortress. The inner membrane's chemistry even resembles a bacterial membrane down to specific lipids you don't find elsewhere in your cells. They divide themselves. You do not build a new mitochondrion from a blueprint. Existing mitochondria grow and pinch in half, on their own schedule, the way bacteria do, which means there is an unbroken chain of cell divisions, running from the original captive bacterium to the mitochondria beating in your heart at this moment. Nothing in that lineage has ever stopped dividing. Not once in 2 billion years. We know the family. Genetic comparison places the mitochondrian's origin before the root of the alpha proteobacteria family tree, a group whose modern members include Rickettsia, the intracellular parasites responsible for typhus and Rocky Mountain spotted fever. Your power plant's nearest wild cousins are microbes that specialize in intense metabolic teamwork. That is not a coincidence. It is a family talent. And your immune system still knows. This is the fingerprint that ought to raise the hair on your arms. If a person suffers massive physical trauma, a crush injury, a bad car accident, cells rupture and their contents spill into the bloodstream, mitochondria included, and the immune system attacks the debris as though it were a bacterial infection. The circular DNA, the distinctive protein fragments. To the ancient pattern recognition machinery of your immune cells, it all reads as bacteria. In severe trauma, this can trigger a body-wide inflammatory storm resembling sepsis in a patient who has no infection at all. Your own body cannot tell your mitochondria apart from an invading microbe. It isn't making a mistake. 3.5 The terms of the deal. Every partnership starts with what each side gets. The bacterium got shelter and a steady supply of food. The host ultimately got something even better. Oxygen respiration, the 15-fold energy bonanza from Chapter 2. The ability to run electrons all the way down onto oxygen and tax the entire descent. But partnerships change. And what began as a deal between equals became something much stranger. The bacterium's genome dissolved. A free-living alpha proteobacterium might carry a few thousand genes. Most of those genes were simply lost, redundant, unnecessary in the shelter of a host. Others were physically relocated, migrating out of the organelle and into the host's nucleus, where they sit to this day, in your chromosomes, still making mitochondrial proteins, but doing it from headquarters. What's left inside a human mitochondrion is 37 genes. 37 out of thousands. Everything else the organelle needs, well over 1,000 different proteins, is manufactured out in the cell under nuclear instructions and imported through dedicated channels in the mitochondrial membrane. Like deliveries through a service entrance. The prisoner cannot leave. It cannot survive alone. It hasn't been able to for 2 billion years. Take a mitochondrion out of a cell and it dies, having long ago surrendered nearly everything it would need to live. Which raises the obvious question. Why keep any genes at all? Why not move all 37 and be done with it? The leading answer is beautifully practical. The genes that stayed behind are almost all genes for the car respiratory machinery, the electron transport chain and the ATP turbine. And that machinery is dangerous. It handles high-energy electrons. And when things go wrong, electrons leak and start reacting with everything nearby. You cannot manage a machine like that from headquarters, waiting on a memo. You need a local governor, standing at the membrane, able to read the situation and adjust production on the spot. Right now, the mitochondrion kept a genome for the same reason a ship keeps a captain even when the shipping company has an office ashore. And of everything it could have kept, it kept the electron transport chain and the ATP synthase, the exact machines that made it worth swallowing. 3.6 What the merger bought. Now, why did this matter so enormously? Why couldn't a bacterium just get bigger and complicated on its own? Because of a wall it cannot climb, and the wall is geometry. A bacterium generates its power on its outer membrane, the proton gradient of chapter 2, pumped across the cell's own skin. But if a cell grows, its volume increases as the cube of its size while its surface area increases only as the square. The interior swells faster than the power plant that has to supply it. A giant bacterium is not a mighty bacterium. It is a starving one, a large body wrapped in an inadequate skin. And there are hard limits to how much genome and machinery it can afford to run. The eukaryotic cell escaped this by cheating. It stopped generating power on its outer surface and moved the power plants inside. Hundreds of them. Thousands. Each mitochondrion is packed with deeply folded internal membrane, and every square nanometer of that folding is bioenergetic surface. The cell effectively carries an enormous amount of power-generating membrane in a small volume, and each unit of it comes with its own little local genome to run it. The biochemists Nick Lane and William Martin have argued that the consequence is a jump in the energy available per gene of tens of thousands of times over. And energy per gene is the thing that matters, because a genome is expensive to maintain, and a big genome is the raw material of novelty. With a bigger budget, a cell can afford to carry thousands of genes it isn't currently using. It can afford to keep experimental copies around. It can afford to build absurd, elaborate, costly structures and see what happens. This argument is forceful, and it is contested. Other biologists have argued the energetic gap is far smaller than claimed and that cell size and other constraints do most of the work. The debate is live and worth watching, but nobody disputes the core of it. After mitochondria, a single cell could afford things it had never been able to afford in 2 billion years of trying. Some sense of what that means in your own body. You turn over roughly your own body weight in ATP every day. Not because you contain that much, you carry only a few ounces of it at any moment, but because every molecule is spent and rebuilt, hundreds or thousands of times a day, by rotating turbines inside domesticated bacteria, and the oxygen. Take a breath. That oxygen crosses into your blood, rides a red cell, slips into your tissues, and the vast majority of it enters a cell, crosses 2 membranes, and arrives at the end of an electron transport chain where it catches the falling electrons, picks up 2 hydrogens, and becomes a molecule of water. Every breath you have ever taken has ended largely inside the engine rooms of these domesticated ancient guests. One last thread. In humans and most animals, mitochondria are passed down in the egg, not the sperm, which means your mitochondrial DNA came from your mother, and hers from her mother, and hers from her mother, in an unbroken maternal chain running back through an unbroken maternal chain of mothers, grandmothers, and great-grandmothers, up past the origin of our species, tracing back through a vast lineage of ancestors to an anciency. That chain has never been broken. If it had been, you wouldn't be here. 3.7 The second swallowing. Then it happened again. Somewhere in the neighborhood of 1.5 billion years ago, a eukaryotic cell, one that already had its mitochondria, already had its nucleus, already had the whole architecture engulfed a cyanobacterium, The cyanobacteria, you'll remember, are the microbes from Chapter 2 who invented oxygen-producing photosynthesis, rusted the oceans, poisoned the world, and froze the planet. Now one of them was captured, and, as before, it wasn't digested. It settled in. It went on doing what it did, catching photons, splitting water, making sugar. But now it did it for a landlord. Its genome shrank. Its genes migrated to the host nucleus. It became an organelle, the chloroplast. From that single capture came the green algae, the red algae, and every land plant that has ever grown. Which means that a leaf is a bag of enslaved bacteria. The green of a summer forest is not the color of plants. It is the color of a pigment invented by cyanobacteria, in cells that plants inherited by theft, and never gave back. And then the story gets genuinely weird, because other eukaryotes, later, came along and ate those eukaryotes, the ones that already had chloroplasts inside them. And once again, the meal was not entirely digested. The predator kept the chloroplast, discarding most of the rest of its prey. This is called secondary endosymbiosis, and it happened more than once. And it is how photosynthesis spread into strange corners of the tree of life like the diatoms and the dinoflagellates. You can see the whole nested history if you count membranes. A plant's chloroplast has 2, bacterium and host. But some algae have chloroplasts wrapped in 3 or 4 membranes, layer inside layer. A set of Russian dolls, in which each shell is the skin of a different organism. And the innermost one is a cyanobacterium eaten a billion years ago. And in 2 obscure groups of algae, there is something more astonishing still. Tucked in between those membranes, alongside the stolen chloroplast, is a nucleomorph, the shrunken, withered nucleus of the eaten eukaryote. It still has chromosomes, 3 of them, tiny. It still has genes. It still gets read. It is the vestigial brain of a creature that was devoured before there were animals, still faintly ticking away inside its devourer's descendants, hundreds of millions of years later. Finally, in case you're inclined to think the original swallowing was a one-in-eternity fluke, a nondescript freshwater amoeba, called Pollinella, did it again. On its own, independently. Roughly 100 million years ago, it took in a cyanobacterium of an entirely different family, and kept it. And that captive is now well down the same road. Genes lost, genome shrunken, dependence set in. The door was never locked. It's just almost never opened. 3.8 Who was the host? For decades we knew the guest and had no idea about the host. There was one very large clue, and it was maddening. The eukaryotic cell is a mosaic. Look at how it handles information, copying DNA, reading genes, building proteins, packaging chromosomes, and it looks archaeal, right down to the details. Look at how it handles metabolism, and how it builds its membranes, and it looks bacterial. We were, unmistakably, a hybrid of 2 domains, and nobody could say who our archaeal parent had been, because nothing remotely like it had ever been found. Then, in 2015, someone went looking in the mud. The place is called Loki's Castle, a field of hydrothermal vents on the Arctic seafloor between Greenland and Norway, named by the Norwegian team that found it. Researchers sifted DNA out of the sediment nearby, assembled the fragments, and pulled out the genome of an archaean that no one had ever seen, cultured, or suspected, Eukaryotic. Its genome was full of things that were not supposed to be there. Relatives of actin, the eukaryotic cytoskeleton protein, membrane-bending proteins, vesicle trafficking machinery, genes that every textbook said were eukaryote only, sitting in an archaean in Arctic mud. Then came the others. Thor, Odin, Heimdall, collectively, the Asgard Archaea, and every new genome carried more of the eukaryotic toolkit in organisms that are not eukaryotes. 2 conclusions followed, and both are large. The first, the host cell did not invent the eukaryotic toolkit after acquiring mitochondria. It already had the beginnings of one. The scaffolding, the membrane shaping, the internal trafficking, the rough draft may have already been taking shape in an archaean before the bacterium ever moved in. Whether these proteins evolved to help the host interact with its neighbors, or exploded in complexity right around the time the merger took place, is a puzzle biologists are still piecing together. Either way, the evolutionary blueprint was deeply rooted in our archaeal parent. The second is a redrawing of the map. In Chapter 2, Carl Weiss gave us 3 domains, bacteria, archaea, eukarya, standing as equal branches. The evidence now increasingly says that's wrong, that eukaryotes are not a third branch beside the archaea, but a twig inside them, growing out from among the Asgards. 2 domains, not 3, which has a consequence you may want to sit down for. You are an archaean, a very strange archaean, one that swallowed a bacterium 2 billion years ago, and built an empire on it, but an archaean nonetheless, on the same branch as the microbes in the mud of an Arctic vent. And then, in 2020, a team in Japan did the thing everyone had assumed was impossible. They grew one. They took sediment from a deep methane seep off the Japanese coast, put it in a bioreactor, and waited. And waited. The bioreactor ran for years. Then came the slow, agonizing work of enrichment in subculture. From first sample to published paper took roughly 12 years. They named it Promethearchaeum centrophicum. It divides about once every 2 to 3 weeks. For scale, a well-fed E. coli divides every 20 minutes. It is small and round, and under the microscope it does something no one expected. It extends long, branching, tentacle-like arms. It cannot engulf anything. It has no phagocytosis. It makes its living by breaking down amino acids, and handing its chemical waste, hydrogen, to partner microbes that consume it. Grown alone, without its partners, it chokes on its own byproducts and stops. It is not a predator. It is dependent. It is half of a relationship, and that suggests an origin story quite different from the one we started with. Not a mouth, an embrace. Picture a slow, patient, tentacled archaean on the ancient seafloor, locked in a metabolic partnership with a bacterium that fed on what it threw away. 2 organisms leaning on each other because each one's garbage was the other's groceries. Picture the archaean's arms reaching around its partner, holding it close, the way it holds its partners today. Picture those arms, over a great span of time, wrapping further and further, until they met and fused behind it. Entangle, engulf, absorb. If that's how it happened, then we did not begin with a predator and prey. We began with 2 creatures who needed each other so badly, for so long, that eventually, there was no line left between them. I want to be honest that this is a model, not a documented history, and the details are argued over hard. But it is what the only cultured relative of our own host lineage actually does, out in the dark, right now. 3.9 The wall around the genome. Eukaryote comes from the Greek eu, true, karyon, kernel or nut. The true kernel, the nucleus, it is the feature the whole domain is named for. Physically, it's a double membrane wrapped around the genome, continuous with the rest of the cell's inner membranes, and punched through with thousands of nuclear pores, huge protein rings, among the largest machines in the cell, functioning as customs checkpoints. Nothing crosses without the right molecular passport. Messages go out, machinery comes in, the border is enforced. How it formed is reasonably clear, most likely by membranes folding inward and around. An idea made much more plausible now that we know the Asgard host already carried membrane-bending equipment. Why it formed is the more interesting question, and one leading answer points straight back at the guest. When the bacterium moved in, its genes streamed into the host's genome. But genes were not all that came across. Bacterial genomes are infested with a kind of molecular parasite, jumping genetic elements, that splice themselves out off an RNA message, after the message has been made. When the endosymbiont's DNA flooded into the host, these elements came with it. They spread, they lodged themselves inside the host's genes, in the middle of them, chopping useful instructions into pieces separated by nonsense. Their descendants are still there. They are called introns, and the great majority of the length of a typical human gene is intron, meaningless filler that must be cut out of the message before it can be read. Now consider the crisis this creates. In a bacterium, ribosomes grab an RNA message and begin building the protein while the message is still being transcribed. It's ruthlessly efficient. But if that message is riddled with introns that must first be snipped out, and snipping is slow, and the ribosome is fast, then the ribosome will seize an unedited message and dutifully manufacture garbage. Every single time. A cell that does this does not survive, unless it builds a wall. Put the DNA in a sealed room. Put the editing machinery in there with it. Keep the ribosomes outside. Let nothing leave until the message has been fully edited and checked. On this hypothesis, proposed by William Martin and Eugene Koonin, the nuclear envelope is a quarantine, thrown up in an emergency. But the vault served other vital purposes, too. A giant cell with a massive genome needs structure. The nucleus provided a heavy, reinforced anchor system, allowing the cell's moving skeleton to pull and tug on the vault to coordinate gene reading without tearing the fragile DNA strands apart. It also provided a control space to manage the highly reactive, damaging chemical exhaust leaking from the new mitochondria, deploying a specialized toolkit to scrub the engine's emissions before they could damage the master blueprint. The nucleus may have begun because the guest brought fleas, but it became the ultimate structural shield. That's a hypothesis, not a fact, and it is not universally accepted. But whatever drove it, the consequences of that wall were immense. By separating the reading of a gene from the building of a protein, the cell created an entirely new floor in the building, a space between instruction and product where the message could be inspected, edited, delayed, exported, or destroyed. Regulation exploded in sophistication, and it opened a trick that would matter enormously later. If you can cut a message into pieces and stitch it back together, you can stitch it back together different ways, and get different proteins out of the same gene. A human has only about 20,000 protein-coding genes, startlingly few, fewer than a grape, which hoards duplicate copies of its DNA, and makes far more proteins than that. The nucleus is where the difference is manufactured. 3.10 The inner city. Beyond the nucleus, the eukaryotic cell built an interior. Extending outward from the nuclear envelope, and continuous with it, is the endoplasmic reticulum, a vast, folded, membranous labyrinth that fills much of the cell. Stretches of it become studded with free-floating ribosomes that dock temporarily onto its surface, giving it a pebbled look under the microscope. This is where proteins destined for export, or for membranes, are threaded inside to be folded. The smoother stretches build lipids, store calcium, and neutralize toxins. From the ER, cargo moves to the Golgi apparatus, a stack of flattened sacks that functions as a combination finishing shop and sorting office. Proteins arrive raw. They are modified, sugars attached, ends trimmed, and then labeled with molecular address tags and dispatched. Some go to the lysosomes, sealed sacks of digestive enzymes, the demolition and recycling yards, where the cell breaks down what it has eaten and what it has worn out. The enzymes are ferocious. They are kept behind a membrane, for the same reason you keep acid in a bottle. And moving between all of these are the vesicles, small bubbles of membrane, that butt off one compartment, travel, and fuse with another, each carrying a molecular label read by matching proteins at the destination. It is a postal service, with zip codes, inside a single cell, too small to see. And here's why it belongs in this chapter rather than a chapter on cell biology. To engulf anything, to reach around something, wrap it, pinch the wrapping shut, and take it inside, you need exactly this, a membrane that bends, buds, seals, and fuses on command. Which puts us in an awkward loop. You need this machinery to take in a mitochondrion, but you arguably need a mitochondrion to afford this machinery. Which came first? The Asgard evidence, and the tentacles of Promethearchium, soften the paradox nicely. The host didn't need the full cathedral. It needed a cheap, modest, low-budget version, enough membrane bending to wrap its arms around a partner it was already living with. The elaborate version, the ER and the Golgi, and the whole logistics empire, could come afterward. Once the power was flowing and the cell could afford to build. You don't need a crane to hold someone's hand. 3.11 The skeleton that walks. The last great piece of the architecture is a skeleton, but not the kind you're picturing. Not bone, not fixed, not passive. A eukaryotic cell cytoskeleton builds itself, dismantles itself, and rebuilds itself somewhere else, continuously, all day, for your whole life. There are 3 systems. Microfilaments, made of actin, are thin and flexible. They give the cell its shape, let it crawl, and pinch it in two when it divides. Much later in this book, in animals, actin will be conscripted into the machinery of muscle, which means the same protein that lets an amoeba ooze is the protein contracting your heart. Intermediate filaments are ropes, tough, tinsel, unglamorous, holding things together under strain. One of them is keratin. Your hair and your fingernails are intermediate filaments, extruded and hardened. Microtubules, made of tubulin, are hollow tubes, stiff, straight, and dynamic, growing and shrinking from their ends. They are the cell's highways, and on those highways there is traffic. Kinesin is a protein with 2 feet. It binds to a microtubule and it walks, swinging one foot past the other, hand over hand, roughly 8 nanometers a step, about 100 steps a second, hauling cargo behind it. Dynein walks the other direction. This is not a figure of speech, and it is not a simplification for beginners. They walk. It has been filmed. Right now, in every cell in your body, there are molecular porters trudging along tracks, carrying freight to addresses. Down the length of your longest nerve cells, which in a tall person can run a meter from spine to toe, cargo is hand-carried by these things, step by step, for weeks. And then there are cilia and flagella. Take 9 pairs of microtubules, arrange them in a precise ring around 2 more in the center, and stud the whole assembly with Dynein motors. When the motors try to walk, and the tubes are anchored so they cannot slide freely, Sliding becomes bending. Bend, release, and you have a whip. Sperm cells swim with one. The cells lining your airways are carpeted with them. Thousands upon thousands, beating in coordinated waves like a wheat field in wind, sweeping mucus and dust up and out of your lungs every minute of your life, including this one. Cilia lining your brain cavities keep your spinal fluid circulating. Cilia and the developing embryo help decide which side of your body your heart goes on. One final note, because it's a lovely piece of evolutionary trivia. The bacterial flagellum from Chapter 2, the rotary motor, the corkscrew propeller spun by protons, and the eukaryotic flagellum you just met are very different structural inventions. One spins, the other whips. While the bacterial version repurposed an ancient molecular pump to assemble its propeller, the eukaryotic whip was built by extending the cell's internal skeleton and membrane-shaping toolkit outward into the sea. Life figured out how to swim twice from scratch. It came up with two totally different designs that work on completely unique engineering principles. 3.12 Sex. There is one more eukaryotic invention, and it is the one nobody has fully explained. Bacteria reproduce by dividing, and they swap genes sideways in the loose, opportunistic way we saw in Chapter 2. Eukaryotes do something formal and strange: meiosis. A cell has its chromosome count to make gametes, egg and sperm in our case, and two gametes fuse to restore the full set. And during the having, matching chromosomes physically line up, cross over, and trade pieces. Notice what that is. It is not reproduction. Asexual reproduction works perfectly well and is simpler and is faster. Sex is not a way of making offspring. It's a way of shuffling, which has been welded onto the making of offspring, and it is spectacularly expensive. If you reproduce asexually, every gene you have goes into every offspring you make. If you reproduce sexually, you throw away half your genome every generation, and you must first find a partner, court a partner, and hope the partner cooperates, all of which costs time, energy, and sometimes your life. On paper, an asexual mutant dropped into a sexual population ought to outbreed it into oblivion within a few dozen generations. And yet essentially every eukaryotic lineage on Earth does it. Even organisms long assumed to be lifelong celibates, Giardia, various amoebae, turn out to carry the full meiotic toolkit in their genomes, quietly, whether or not we've caught them using it. Meiosis appears to have been present in the common ancestor of all eukaryotes. Invented once, very early, never abandoned, which means it must be paying for itself hugely. The leading explanations: recombination combines good ideas. Two beneficial mutations arising in two different individuals can be brought together into one genome. Without sex, they can never meet. Each lineage has to invent everything itself. Sex reverses a ratchet. In a lineage that never recombines, mildly harmful mutations accumulate and can never be cleanly removed. A one-way slide called Muller's ratchet. Sex lets two parents produce a child cleaner than either of them. Sex is a moving target. Parasites evolve fast, and they adapt to whatever is common. A genome shuffled fresh each generation is much harder to lock onto than one that never changes. As for where meiosis came from, the best guess is that it was repurposed from the machinery of DNA repair. The enzymes that carry out meiotic crossing over are close relatives of the enzymes that mend a broken chromosome by copying from an intact one. Sex may have begun as first aid, and only later become mixing. But the honest summary is that the maintenance of sex remains one of the great unresolved questions in evolutionary biology. We are not sure why we do this. The question is being asked, of course, by organisms who are themselves the product of it. 3.13 The cell that could afford to be complicated. Step back and count what one lineage acquired. An energy supply beyond anything the world had seen. A walled, guarded, editable genome. An internal factory with a delivery network. A self-building skeleton with motors that walk. And a system for shuffling genes. Every one of those is machinery that multicellular life would later borrow and specialize. A neuron is a eukaryotic cell that stretched its cytoskeleton into a cable a meter long. A muscle fiber is a eukaryotic cell that turned actin into an engine. A white blood cell chasing down a bacterium in your bloodstream right now is doing so by engulfing it, using, essentially unchanged, the very trick that started this whole story 2 billion years ago. The toolkit was complete. Everything that would build a fern, a fly, a shark, or a mind was, in some form, already sitting in that first eukaryotic cell. And nothing happened. For something like 1 billion years after eukaryotes appeared, the world remained a place of single cells. The eukaryotes diversified, certainly. They became amoebae, and ciliates, and algae, and a vast unnamed multitude, but they stayed alone. Most of them still are. Open a drop of pond water, and the overwhelming majority of eukaryotes you'll find are single cells, complete unto themselves, with a nucleus and the mitochondria, and the whole cathedral, and no interest whatsoever in cooperation. They have been doing perfectly well like this for longer than animals have existed. So let's be careful about the story we tell. The eukaryotic cell did not cause complexity. It did not make multicellular life inevitable. It made it affordable. It stocked the workshop, and paid the power bill, and left the door unlocked. Someone still had to walk in and build something. And then, in seaweeds and in fungi, and in animals, independently, in lineage after lineage that had no idea what the others were doing, cells began to divide, and not let go. That is chapter 4.